Welcome to the Biognosys webinar, the quantitative proteophenotype measurements and biological significance. My name is Ernst Hafen. I'm your moderator today. I'm a professor at the Institute for Molecular Systems Biology at ETH Zurich, and it's a great pleasure to introduce our two speakers. First, we will have a short introduction on Biognosys by its CEO and co-founder, Oliver Rinner. Oliver studied biochemistry and psychology at the University of Tübingen, then did his PhD at the ETH Zurich and the Institute of Brain Research at the University of Zurich, and did a postdoc with Ruedi Aebersold at the Institute of Molecular Systems Biology, and in 2008 he founded Biognosys and has been the CEO since then. He will shortly introduce Biognosys, but before he does that, I will also tell you about our main speaker today, that is Ruedi Aebersold, who really needs little introduction. He is a founding father of the field of proteomics, and he started his career in Basel, then moved to Caltech, from Caltech to Seattle, and then to Canada, back to Seattle, where he co-founded the Institute of Systems Biology, the first institute with such name. And we were then very happy around 2000 to recruit him back to Zurich, to the ETH Zurich, where he then founded the Institute of Molecular Systems Biology, and where he has made fundamental impacts and contributions to the field of proteomics, which is also the title today, quantitative proteotype, because personalized medicine has largely focused on the genotype, but much more important and much more difficult to describe is the phenotype, the symptoms, and how we can treat the symptoms, and that is the phenotype. And quantitative proteomics is a way to tackle this problem. So without further ado, I'd like to hand over to Oliver Rinner to tell us a bit about Biognosys, and then we'll move to Ruedi Aebersold. I should also say, before I hand over, that please ask questions during the seminar. So at the end of the Ruedi's presentation, we will answer these questions. Type them into the chat box, and we'll record them and answer them later. So now, Oliver, please.
Thank you, Ernst, and thank you, Ruedi. So I just want to give you a very short introduction, because then we want to move over to the presentation of Ruedi. So Biognosys, our mission really is enabling researchers to do better biology, to better understand their biology by providing them the technology to analyze the proteome. And we do this in two ways. So either people don't have access to a mass spectrometer, so we provide them with contract research services, so do the projects for them. Or for the experts, we have software and reagent products that enable them to do this new way of proteomics themselves. So originally, we spun off from the lab of Ruedi Aebersold here, very close, actually exactly where I'm sitting now, at the ETH Zurich. And since then, we have been inventing, developing, and providing cutting-edge proteomics for the R&D market. That's really our focus, to support the R&D in the field. We are still close to Zurich, with our facility in Schlieren, close to Zurich, and also in Beverly, USA. And now it's almost 10 years that we have been working with customers. And what we learned from our customers over the years is that in the end, technology serves to answer biological questions. So it's really about the biology, but in order to do that, the technology certainly needs to be able to deliver that. And I think it's fair to say that until recently, or not too long ago, the perception of proteomics was that either you could kind of look at many proteins, but it was lacking the throughput, sensitivity, precision, validation in the eyes of many researchers. So that they had to rely on antibody-based methods, which in turn certainly did not allow you to look at many proteins at the same time. Now this has changed with this new way of doing proteomics that some people call Nextgen proteomics, or technically data-independent acquisition. You really can have both depth, actually much higher depth than ever before, but I think equally important, the reproducibility and precision to do a better proteomics and actually to do a better biology. And that's our focus today. So let me just close the technology part with a referral to our newspaper just published now, I think this week in MCP, where we really try to bring everything together that we in the field has developed over the past years in terms of acquisition technology, bioinformatics, statistics, to enable now a deeper and better view than ever on the proteome. And we are happy to provide you with a reprint. Please ask us in the end. You can also find it yourself at the newest MCP online. So the message in short, this new way of proteomics, SWAF, DRA, whatever it is called, enables you to acquire the data in a parallel way. Before it was sequential. That means you can now identify in reality more proteins than you could even do theoretically before. And if you go to really complex proteomes, so in panel C, we have such a mixed proteome, we can go beyond 10,000 protein identification, single injection, really showing the capacity and the depth that the technology can now achieve. So I think it's already, and we do this already with our customers, to tackle the biological questions, and now I'm looking very much forward to hear from Ruedi what he has to show us about the newest research in that field. Thank you. So I'm just changed now to presenter. Good.
Okay, thank you, Ernst and Oliver, for the introduction and the opportunity to talk here. I was asked by Oliver to speak not so much on technical aspects. It is of course exciting to see that the field of acquisition and data processing is rapidly advancing in this DIA, SWOS, or hyperreaction monitoring field. These are basically synonyms for the same thing. And Oliver asked me to more talk about the biological concepts and significance of the results that we obtain by these techniques. So if we look at the current status of the proteome analysis in the human proteome case, similar situation would be for other proteomes. We can see that a large number of peptides have been identified. In fact, this slide shows a printout from a peptide atlas database that we created in Seattle and is managed right now by Eric Deutsch at the ISB in Seattle. And it shows a tremendous increase of spectra, fragment ion spectra, that have been generated from the human proteome. These peptides, fragment ion spectra, are then assigned to distinct peptides, and currently somewhat around 1 million distinct human peptides have been sequenced. We can now ask, what does this actually mean for the proteome? And here we list again, courtesy of Eric Deutsch, a saturation curve of the discovery of the proteome using classical DDA-based tandem mass spectrometry techniques. We see on the blue side, in the blue panel here, that over the last few years, very large data sets were being generated, and they led to an increase, steady increase, and eventually saturation of the peptides identified, roughly at 1 million peptides. We see on the right-hand panel, we see that this the addition of these very large data sets did not really discover any more proteins, at least not many, provided that the false discovery rate of these inferred proteins was correctly estimated. So the current situation of proteomics based on mass spectrometry and the human proteome would be roughly that the human proteome has been discovered to saturation but not to completeness. We can continue to, of course, increase the completeness of proteome coverage, but that's not what we're talking about. We also know that the proteome and the transcriptome do not contain the same biological information. In fact, there was a webinar a short while ago from Andreas Bayer, who investigated together with us this question in very great depth, and we published a review, and Andreas explained the rationale of this statement that they're not the same biological information is represented by mRNA and protein. Then the third point here is that for the most part, proteomics so far has treated the proteins largely as independent molecules. Basically the same way, or in analogy to transcripts, but this is not how proteins function. Proteins function for the most part as social molecules, and they operate in the context of functional modules, protein complexes or networks, and we think that the social networks of proteins is in terms of how they interact with other elements of the cell is an extremely rich source in biological information. So why do we think that? We can go back in the old literature, 60, 70-year-old literature, and we see an extremely interesting and pioneering example which indicated that it is not just the abundance of a protein, but the way proteins are organized in a module or in a complex that determines a biological function all the way towards a clinical phenotype. The example is sickle cell anemia, and this was work from Linus Pauling in the late 1940s, and he defined sickle cell anemia as a molecular disease. So this really provides a paradigm that is still valid, is not really that widely used, at least not in the omics field, and the paradigm basically says the following. If we have a mutation in a specific locus in the genome, that this mutation may, as is in the case in sickle cell anemia, introduce a single amino acid change in the sequence of a protein, and that this protein, when it's expressed and is part of a protein complex, in this case the tetrameric form of a hemoglobin, that this mutation may lead to a change not only in the abundance or the function, but also in the way the protein is organized structurally in its complex. And this complex, the single amino acid exchange, in the case of sickle cell anemia, created a different structure of tetrameric hemoglobin, which is linked very tightly to the emergence of a disease, sickle cell anemia. So this established effectively that a mutation can change or disrupt a structure of a complex, and that this leads to a phenotype. So this is the paradigm we would like to continue to pursue at the level of the proteome using mass spectrometric techniques, and we extend, therefore it's our goal to extend Pauling's principle to the age of the proteome. What we effectively would postulate is a term, the proteotype, and this proteotype we define as the acute state of the proteome in a cell, and we're not talking just about the independent proteins as they are present in the cell, but also their organization in modules and complexes. So this is of course an enormous task. We just managed roughly to, so far in the field of proteomics, to quantify and identify most of the protein components in a cell, and it is now the task for the future to also learn how are they connected and how is the level of connectivity change under certain conditions. For instance, if the genotype is varied through mutations, copy number variations and so on, or if the cell is exposed to environmental or other changes. And the other question would be, how is this altered proteotype affecting a phenotype exactly as it is exactly as it was done by Linus Pauling in the context of a specific complex. So critical questions that we would like to eventually answer would be, to what extent are cellular proteins organized in macromolecular structures? To what extent is the genotype determining this proteotype, the abundance as well as the organization of the proteome? Does the phenotype classify groups? Does it form like biomarkers that we can use to, for instance, distinguish healthy from diseased tissue? Does the proteome context reveal information that is not apparent simply from quantitative protein measurements? So basically making the transition from the thinking how transcripts are being treated as independent units to the treatment of proteins as functional modules. And then we would like to ask, do changes in the modular organization of the proteotype have functional consequences as was shown in a specific case by Linus Pauling. We believe that the underlying necessity to answer these questions is a new type of data representation, and that is not a list of proteins identified, but a data matrix as shown here, that where a number of analytes, in this case proteins or proteoforms, would be quantified over a large number of samples. So we're not interested so much in lists like inventory of proteins in a cell, but a data matrix where a large number of proteins, ideally all proteins, would be quantified precisely across a large number of samples. This could be in the context of clinical questions or in systems biology or other biological questions. So when I say the new currency of proteomics, this data matrix is essentially the result of the highly reproducible and relatively quick and deep measurement of proteins across cohorts, and this is uniquely and highly effectively achieved by SWOS, DIA, or hyperreaction monitoring, these massively parallel targeting techniques that were developed and initially exemplified by SWOS mass spectrometry. So if we take as the currency of our information to infer biological information this matrix, we can ask of proteins measured across conditions. We would like to have optimally a matrix where we would quantify every protein in a sample and do this over a very large number of samples. So we cannot really claim that we can quantify every protein, even though the number of proteins that can be quantified in a single injection has been steadily and quite rapidly increasing, and Oliver just indicated the last results from Biognosys, where they reach 10,000 proteins, which is a remarkable number. But I would like to say that even if the number of proteins is somewhat lower, we can already acquire highly significant biological results. But we would like to ask now, and this will be the three topics I'm trying to explain in the remainder of the talk, three fundamental questions about this data matrix. So the data matrix is the center of our attention, and I'd like to answer and examine and answer three questions. The first one is, what are the optimal dimensions of this matrix? Should we spend more effort on going very deep, or should we spend more effort and go very broad in terms of samples? Secondly, I would like then to answer or examine what is the most informative subset of proteins in the matrix. In other words, do all proteins that are present in this matrix have the same biological information, or are there some that have to tell us a stronger message and others have nothing to really specific to tell us? The third question I will try to answer if you have time. Can we infer information about the organization of proteins in the cell from the data represented in such a data matrix? So these are the three short vignettes that I'm going to examine. The first one has implications for the design of biologically relevant or clinically relevant experiments. The second and the third are directly biologically relevant questions. So I'll try to discuss these three questions in the context of biomarker studies, where we have a scenario where we have control and disease state, but it should be, I would like to make the point very strongly that exactly the same arguments would apply to genome-wide or proteome-wide association studies, identification of clusters behaving similarly under different conditions, for machine learning, network inference, and for other types of analysis. So I'm just choosing biomarker study because it's very plausible and everyone understands what the goal of such a biomarker study in principle is, to separate different groups, let's say control and disease. But it is only one example of the questions that can be addressed by this approach. So why protein biomarkers? It is, of course, a highly relevant question. Hundreds of thousands of research papers have been published, and most papers are limited to a relatively small cohort and limited number of proteins that have been analyzed, for instance, by mass spectrometry or also by antibody-based techniques. So we might ask, what are the relevant parameters for successful biomarker classification projects? And this is the first issue related to this data matrix. We'd like to ask, what are the optimal dimensions of this matrix, where if we had certain 100% of effort to spend, would we spend it on going very deep into the proteome by, for instance, extensive fractionation of samples, or would we go broad by analyzing a certain high number of samples at a certain depth? That's a question I'd like now to answer, and I'll do this in the context of a study that was done with Ting Wang, who is a statistical graduate student in the group of Olga Witek, and it's been done in collaboration with our group. I might also at this point out that a lot of the data I'm showing are not published, so I would hope that the slides will not be widely distributed. This is for information only. I am confident that we will publish this relatively soon, but nevertheless I would like to ask to respect the fact that the data are not published yet. So what are we trying to do here? We have a data matrix here, exemplified by a training cohort, where we have a certain number of proteins quantified over a certain number of subjects. And now we would like to ask, if we go to find out whether these data separate two groups, case and control, very objectively, if we went to a secondary cohort, a validation cohort, should we include more subjects or should we attempt to identify more proteins? And we do this by basically, or Ting did this by developing a mathematical model to simulate the behavior of biomarker discovery and validation experiments in a computer, making certain assumptions about the protein behavior in different mass spectrometric techniques. And the mass spectrometric techniques she assumed was DDA analysis by spectral counts. The data that are the basis for that is shown here in this Nature paper. The second one is SRM, space targeting, is represented by data in the EMBO molecular medicine paper. And the third is SWOS data on a manuscript that's under review, and these are SWOS data from a number of cell lines. What she then did, she plotted the noise behavior of the data generated by the different methods in a plot where we have basically standard deviation of variability of the protein observed as a function of the abundance of the protein. And when we go here over to the right-hand side, which is the SWOS DAA data, we see that the behavior, the noise behavior, follows the very classical trumpet plot that has also been always observed by, for instance, transcript profiling data, that proteins which have a high, or objects that have a high signal-to-noise ratio, have low variability, technical variability, and objects observed with a low signal-to-noise, if the noise level goes up and the standard deviation or the variability is high. It's a very classical distribution, and we see that the SRM data do not follow this distribution because these proteins were highly selected for those proteins which behave well and the assays were optimized. And we also see that in the TCGA data on spectral counting, this shows an unusual behavior, that it starts out like a trumpet plot and then the variability goes down. We don't then actually understand that behavior. But anyway, each one of these three different papers had a certain noise behavior, and now into this noise behavior she modeled she modeled potential biomarkers. So basically, the proteins here exemplified as representing SWOS data are separated into 5 quintiles of different abundance based on signal intensity, and into each one of these quintiles, Ting in a computer simulates 3 biomarkers which separate the protein in the case of control and sample by, let's say, a factor of 2-fold. And this leads to various data sets that can be simulated in a computer, namely proteins which are relatively few proteins, which are very precisely measured, as it would be in, for instance, SRM experiments, proteins of intermediate strength of signal intensity, which have a moderate level of variability, and then proteins of lower signal intensity, but of course a larger number of those, but they have larger variability. So then Ting basically does hundreds of simulations, and she chooses various numbers of proteins with the corresponding noise behavior, different numbers of biological replicates, so it would be the number of cases and control, and then she basically asks which ones of these simulations have the highest propensity to separate cases and controls, assuming the background noise behavior or signal behavior of the specific technique, and assuming increasing number of cohort size and increasing number of proteins quantified. So the results are actually extremely interesting. So we see here the results of these simulations. And I would just, I want to go quite quickly through that, and basically good separation means solid color here, and the number of 1 means a high predictive accuracy or a high sensitivity of identifying predictive proteins. We see the x-axis is the number of increased proteins, and the y-axis is the number of increased subjects from bottom to top. So it is not surprising that if we have only like 10 cases and controls in our sample, that we do not reach perfect separation, because the study is simply underpowered. But if we go up in the y-axis, we see that the separation becomes very good to almost perfect under these simulated conditions. We also see that if we increase the number of proteins that enter the study, that we see a counterintuitive or paradoxical behavior, in that initially the separation is quite high, or the mean predictive accuracy, but then the more proteins we add, the lower it gets. We see this also by the mean sensitivity of identifying predictive biomarkers. We see that initially with relatively few proteins it's relatively high. The more proteins we add, the worse it gets. So what does this mean? This basically means that it is not the number of proteins that indicates the behavior and the ability to use the data matrix for predictive purposes. It is the quality of the data, and specifically the noise behavior of the protein. So if we keep adding proteins, large numbers of proteins that have unfavorable noise behavior, we actually dilute out the signal that separates the samples apart, but we dilute it essentially in noise. So this leads me to the first conclusion of these simulation experiments. More proteins decrease the predictive accuracy and sensitivity of identifying predictive spots, because more protein inevitably means that the noisier proteins are entering the separation of the case and control. More subjects in every case increase the accuracy and sensitivity, and the conclusion from the experimental point of view would be that if we want to retrieve in biologically relevant or clinically relevant information, the experimental techniques should optimize throughput, of proteotype measurements, should optimize for low technical variability and high reproducibility across the cohort, and this is exactly the strength of the SWOS DIA technique. So we believe that these DIA techniques are ideally suited for cohort studies, because they provide a relatively large number of proteins at relatively low technical variability, and it will be prudent to sort out the highly variable proteins before they enter the analysis. So now the second question I'd like to ask is, what is the optimal set of proteins to reach a biological clinical conclusions? We are obviously able to measure now thousands of proteins across hundreds of conditions, but we do not really know a priori whether every protein has the same capacity to tell us something biologically important. So this is the question we'd like to now address. The research question is, are some proteins more informative than others? For instance, for classification or biomarker research, but this extends again to all the other aspects that can be addressed with proteomic data. The study goal here was to quantify the variability of plasma proteotype in terms of the routes of variation. Is it genomically variant? Is it a variable induced by environmental or longitudinal effects? And then quantify the relative contribution to the variability observed based on the where the variability came from, whether it came from genomics, from the environment, or from some other factors. The study design was, this is actually the only data that I showed that I published. This is a paper from Yansheng Lu, postdoc here about 2 years ago, and he generated the plasma proteome maps of a human twin cohort. He mathematically deconvoluted the roots of the observed biological variability, and then he associated genetically determined variation with the causing locus, so-called pQTL mapping, which I'm not going to show for reason of time. So why twins? Twins are extremely interesting cohorts because we have two classes of twins, identical or monozygotic twins, which are genetically identical, and dizygotic twins, which are not genetically identical. So we would assume that the variability observed in monozygotic twins or dizygotic twins may be different because in one case the two twins are genetically the same, and if they differ in some form of phenotype measured, this would be likely induced by the environmental or other factors, whereas the dizygotic twins are also genetically different, so they will be the variability will be arising from different genotype as well as from different environment. And so formalism has been developed over time by the genetics community, which uses this effect that I just described to quantify the degree of heritability that determines a particular trait, for instance, the abundance of a protein, as we would measure it by mass spectrometry. So we would simply correlate the abundance of a protein or a number of proteins. So each dot here, blue dot, is the abundance measured for a specific protein in either one of the twins of a pair of twins. And if we correlate these abundances for the monozygotic twins, we would assume that the correlation is stronger than the corresponding correlation in dizygotic twins, simply because they're genetically identical and these over here are not. And formalism has been developed, which I don't want to get into, which allows one then, based on a mixture model, to assign basically to partition out the total observed variability into genetically rooted variability, environmentally rooted variability, unexplained variability, and variability that is, for instance, longitudinally occurring if the cohort is structured accordingly. So this is exactly what Yansheng Lu did. He took 36 monozygotic twin pairs and 22 dizygotic twin pairs, and he obtained from Tim Spector in London, who maintains a unique twin cohort database and sample base, plasma samples which were taken from these individuals 2 to 7 years apart. He analyzed these proteins without any enrichment by swath mass spectrometry, and then he used the formalism that I have shown before to deconvolute the variability, source of variability observed. And so this was a total of 232 swath maps of human plasma, and this is effectively the result of this analysis. So this would be on the x-axis the cohort number, this was run in duplicate, and the y-axis the number of proteins. The number of proteins, of course, not very high, not nearly as high as what Oliver explained just at the beginning. The reason, of course, is that this is effectively unfractionated plasma, which everyone who works in the proteomic field knows is a very challenging sample. But these 342 proteins were measured with virtually no missing values over more than 230 samples. Okay, so now if we apply to this data the formalism that I explained, we see that in this graph we basically proportion the source of variability observed to genetics, environment, or longitudinal effects, and this is color-coded here. We see on the left-hand side proteins which have a very strong heritability. That means that the genetics determine very strongly the abundance of these proteins. We see the yellow proteins, on the other hand, which change in their abundance relatively strongly over the cohort with increasing age. And we see proteins which have a very strong environmental effect, for instance, some effect from the lifestyle. So the conclusion from this slide here is that virtually every protein has a different reason, a different overall variability, and also different proportions from the different routes of variability that contribute to the overall picture. So every protein is not the same in its behavior in the human body. So one of the interesting observations we made, which is something that certainly should be considered in biomarker studies, is that over time the genetic control of protein abundance decreases. So we have here the time point 1 heritability component, and then in time point 2, and we can see that this statistically highly significantly lower. So the genetic control of the protein abundance as expressed by the variability observed over this twin cohort is decreasing with age, and that's certainly an interesting observation. If we want to define biomarkers for specific diseases, it matters obviously whether a protein is measured or discovered in a cohort of relatively young people or of relatively old people, because the contributions to the variability are very different in these two age groups. We also asked why some of the protein biomarkers that have been published in papers like papers shown down here never made it through into the clinic. And so this is a common phenomenon, and it's discussed a lot in the proteomics community, that dozens or hundreds of papers are being published where authors use a relatively small cohort to identify biomarker candidates, but they never make it further into the clinic because they somehow fizzle out during validation. And here we have a number of proteins which were postulated or published as potential biomarkers, which were also quantified by Yansheng Lu in this twin cohort. And we see that these proteins all have in common that they have a very strong longitudinal component in their variability and a relatively low heritability component. So that means their abundance variation is strongly affected by the age and to some extent by the environment, but not so much by the genetics. This basically means that these proteins are generally relatively variable in a human population of different age and of different environmental conditions, and so they will simply be too variable to serve as viable biomarkers in a larger population. And the main reason they were identified initially as potential biomarkers simply because there was probably a bias, or the study was undersampled and they appeared with a reasonable p-value, but in a larger powered study they would not be discovered. So it matters for biomarker studies, it matters greatly to see what is the variability of the proteins in a cohort and where is this variability coming from, because we can see then that the exactly opposite behavior is present in FDA cleared and approved plasma proteins, some of which were also observed in this biomarker study, or in these twin cohort studies. So we have 42 proteins which were measured in this cohort, which were also approved biomarkers. We can see that these proteins have generally a higher degree of heritability, that is shown over here, than other proteins that were also measured and in the cohort. So we would conclude that if we want to discover biomarkers in plasma, but by implication probably also in other tissues, that we would need to focus on proteins which have a strong degree of heritability, relatively low variability in the cohort, and only then do we have a reasonable chance to actually use these proteins as definitive biomarkers in in a larger population. So the summary of this part would be the quantitative protein measurement in the twin cohort identifies proteome abundance variability and its roots. We should take into consideration the source of variation, and we see very strong longitudinal effects, and these insights can only be generated by cohort studies where a certain number, a relatively large number of proteins for proteomic studies can actually be quantitatively and reproducibly measured. Now the third question I would like to answer or address is whether we can infer from this data matrix we have now discussed quite extensively issues about the organization of the proteome. We would now like to make the step from using proteins as independent agents that independently of each other operate in the cell to their operation in the form of complexes or functional modules. So the research question here is, can such proteome matrices be used to infer changes in proteome organization, and can this be used as indicators of cell or tissue state, that is, as biomarkers of a different type, not just by the abundance of the proteins. The experimental design was to quantify thousands of proteins by DIA in sample cohorts, and then to mathematically correlate complex component covariance with the phenotype. So I explain what I mean with that. This is the abstract, graphical abstract of the study. So this has to do with prostate cancer, could of course be any other condition. And again, we want to separate groups of healthy and control and different stages of this cancer. This is the actual tissue sample sections from prostate cancer patients, and we can see that the pathologist, which is Peter Wild from the University Hospital in Zurich, here a collaborator of ours, has marked different regions in these sections. Some regions are healthy tissue and some are tissue that is tumorous tissue at various stages of the tissue. So we had 39 patients in this study. He identified 105 tumor regions, and we measured the proteotype of these 105 tumor regions in duplicate. So this would be 210 proteomes. This is the data, basically 210 swath runs of 1 millimeter tissue punches, and we identified consistently about 3,800 proteins across this tissue sample. So that's the data matrix we work with, and classically we now associate each protein with either disease or health, and we would treat them as independent agents. Now we would like to make the step to the organization of the protein, and this is work from Maria Bullian, who is a postdoc here, and she uses prior information on protein complexes, for instance, quorum or reactome or APMS data. So this is established protein interaction information, and she basically asks the question: if two proteins interact in a cell, therefore they're part of a complex, do they concurrently go up or down in each sample measured, which you would expect if the complex is constant and stable, or do they not co-vary, so one goes up and the other one does not, which would indicate that this complex has been disrupted. So this is what she's doing. She takes basically and forms a hypothesis, this red and this blue protein, which we know to be interact. For instance, they define a complex in quorum. She then goes and calculates the variability of covariance of these two proteins in the, let's say, healthy population, the tissue that has been identified by the pathologist as benign, and she asks, do these same two proteins covary in their abundance in the disease cohort differently than in the benign cohort? And this would mean, like, protein may be out here on this protein ratio score, and this would mean that the complex has been disrupted. And then there's statistics to make sure that this is actually true and has substantial statistical power. And in that result, in that particular study, she compared about 25,000 protein pairs that are known to be in protein complexes, and about 350 of those have a significant difference in covariation in the healthy as in the tumor-affected protein samples. So now she can generate a list of proteins, of protein pairs in this case. They're known to be interacting under some conditions, and we ask, and these are now, the score in here indicates a rank score. The red score indicates a score, how far this covariance is outside of the null distribution, the green distribution in the previous slide, and basically indicates that the higher this red score is, the more likely it is that the complex is disrupted in the tumor compared to the healthy tissue of exactly the same individual. And so this will be the proteins that are disrupted. It's the name of proteins, and we would like to show here that these proteins have individual weights and of course they have combined weight. That is the weight. The red score is the combined weight based on the discordance of the variants, and the scores here would be 4 and 1045. This would mean that the protein HSPD1 would have by itself a relatively high degree of separation power of case and control, but the second protein has virtually no power. It would be the 1045th ranked protein that basically has no separation power, but together they come up very high. We also have proteins here, like this one, the case 14, where both proteins by itself would have no value, I mean very, very low discriminant power, but together they come up very high. And one example I would just like in the last minute to explain very briefly, because it actually is medically, biochemically supported. This is the protein LDH, lactate dehydrogenase A, and lactate dehydrogenase B. Neither of these proteins, based on their abundance, has a lot of weight. Together, they have a very high score and are clearly distinguishable in the tumor. What is lactate dehydrogenase doing? It's an enzyme that converts pyruvate into lactate, and it comes in several forms. Basically, it's a tetrameric protein. One form, the LDHA form, and the LDHB form have different catalytic constants. One is very fast, and one is very slow in converting pyruvate to lactate. So you can imagine, if you have the fast form, then the pyruvate is basically converted to lactate. If you have the slow form, then this is slow, and the pyruvate is shunted into the TCA cycle. So this is effectively the area where the very, very well-known Warburg effect in cancer is happening, and this is a key enzyme that is mediating this effect. So we basically observe, based on unbiased, data-driven covariance analysis, that in the tumor and in normal tissue from the same patients, the enzyme here is complex, a tetrameric complex, is composed out of different subunits that translates into fast or slow biochemical processing of the substrate, and this is clearly associated with a well-known cancer metabolic pathway, which was discovered here simply by big data analysis using DIA data, and we can conclude that a mechanistically supported difference in between the healthy and the tissue, the healthy and the tumor tissue of this patient was discovered. And it's of course not the only discovery made. We have a whole range of more than 100 other protein complexes which also help us to classify, and we see that this LDHA to LDHB ratio clearly separates normal from tumor samples, and it also tracks with the difference in their stoichiometry, also clearly tracks with the grade of from low to medium to high grade of prostate cancer tissues in that cohort. So with this, to summarize this part, I'd like to show that we now make the step from analyzing proteins in these data matrices as independent agents to try to document or infer their behavior in functional modules. And we see that we gain new insights into activity of cellular processes and the enzymes that support them, that protein complex subunits with perturbed quantity ratios in prostate cancer have a strong link to disease-relevant processes. And we think that this is a new form of biomarker research where we not just follow the individual protein and its abundance, but also we infer the wiring, basically the organization of these proteins in functional modules in the cell, and we think this is a particularly powerful and useful path forward in the future of proteomics, because it now gets to a level of organization which is simply not apparent from genetic means. So finally, I would like to acknowledge my co-workers whose work I presented here. The SWOS technique, which is the DIA implementation that we developed, was done by Ludovic Gia and Pedro Navarro initially, together with scientists from AB SciEx, Steve Tate and Ron Bonner, and then other members of our lab joined the effort, Ben Collins, Yansheng Lu and George Rosenberger, who did a lot of the software, developed a lot of the software modules that we use. This PCT SWOS analysis was the technique to actually generate these samples, these data matrices from relatively small tissue samples, was the work from Tianan Guo. The prostate cancer project that I briefly showed was the work from Tianan Guo, who generated data, and Maria Bouyan, who developed the computational backdrop of the project, and Peter Wild provided the samples. The biomarker study statistical model is the work from Ting Wan in the group of Olga Witek in Northeastern University in Boston, and the twin study is the work essentially of Yansheng Lu when he got samples from Tim Spector, and Andreas Bayer and Manolis Dermitsakis helped us with the formalism of these twin studies to deconvolute the sources of variation. So with that I would like to stop, and I'm happy to answer some questions.
Thank you very much, Ruedi, for this very interesting presentation showing us how quantitative proteomics and proteotyping can actually help us to get a new view and a quantitative view on the phenotype, and reminding us again that whether it is disease, whether it is individuals in health, natural selection and evolution has always focused on the phenotype and never on single proteins or even single genes. It is always the phenotype, and these technologies now make it possible to address this phenotype in a quantitative manner, and you've shown us in 3 vignettes how this has really helped us to get a new view on cancer or on biomarkers that in the twin studies change longitudinally, and what aspects we have to focus on to make more reliable biomarkers and predictive biomarkers. So I think we've received a number of questions, and I would like to read these now, and maybe you can address this. This is actually from Sonali Mohan. Hello, Sonali. What are the predictive proteins? And also, if number of proteins result in low sensitivity and more variability in smaller cohorts, how does SWOS DA overcome this issue as it acquires the data on all proteins?
Yes, so I think this is actually a strength of the method, because the whole concept behind the SWOS DAA method is conceptually very similar to SRM. I mean, it's also how the software tools, at least the initial software tools, analyzed the data. Is you make certain, basically you create definitive measurement assays consisting of a number of peptides and fragment ion transitions for each peptide, and then this is used to make a statement about the behavior of a protein. In SRM, we have to identify the proteins we want to measure before, and we have to develop these assays, and that was apparent from this simulation experiment where the SRM data set was the one which did not follow this trumpet plot, because the proteins were before qualified to be good good candidates for this particular experiment. In the SWOS measurement, we do not need to know this a priori. We can basically measure a fairly large number of proteins, and then we can use post-acquisition to see, for instance, which proteins are particularly variable over a cohort, and therefore, or a normal population, basically sort them out. So we can basically use the data to determine the abundance of the protein, the variability behavior of the protein. A protein that would be in this population tested extremely variable, we would simply say this is unlikely to be a biomarker, because if it's all over the place, we simply will not. It's very unlikely that we will have that this protein will be able to separate groups. So I think the advantage of the technique is that we can test the behavior of a very large number now from tissue samples, thousands of proteins. We can do some analysis which ones are likely to be informative based on their behavior, and then use these proteins to classify, avoiding the trap that if we just keep up adding proteins which are very, very noisy to our data set, that they might dilute out a signal that is actually present in the data, but is simply drowned out or diluted out by very noisy data. So I think it is advisable to do as many proteins as possible, and then to observe their behavior, and then to select an informative cohort.
Okay, thank you, Ruedi. We can take two questions together. This one is from Natalie Cohen. She wants to know where one can read more about the covariance analysis conducted with protein pairs across the phenotype. And then Praveen Kumar asks more extensively, what is the reason to focus on protein pairs? How about looking at more multi-protein complexes in the future?
Yes. Okay, so you cannot read about that yet. So this is one of the things which is not published, and I thought this would be nevertheless interesting in the context of the question that Oliver posed, and that is, can we extract biologically relevant information from these data sets? And I think this is highly biologically relevant information, as I try to show with this example. So right now we cannot read it. Hopefully soon we're writing this up. And the second question was, why focus on binaries and not on large complexes? So we do actually focus on large complexes in that we do every conceivable binary interaction as covariance, and then we generate a composite score for a larger complex based on the composition of the binaries.
Okay, then there is a question from Pratik Yaktab about the prototype would also have information about post-translational modifications and their effect on organization. Any studies performed or plans to test this on post-translational modification of proteins? Yes,
so this is also a very good question, and it is very clear that variants of proteins, variants in the proteoforms, will likely have an effect on the organization of the proteins. So this could be phosphorylation, this could be splice forms, this could be other modifications. So we recently developed, and it just came out in about a month ago in Nature Biotechnology, an algorithm called IPF, Inference of Proteoforms, and this is addressing the detection of different peptides in DIA data sets. We have also been working on, and I could talk about that at HUPO, that we use a combination of size exclusion chromatography and swath mass spectrometry to basically generate DIA maps of every fraction of a size separation of native complexes, and then since we generate these maps from every consecutive fraction, we can track through the behavior of specific proteins or proteoforms as they associate with complexes that are separable by size exclusion chromatography. And so this now works extremely well, and is also not published, but we're writing this up now, and there we can clearly show that phosphorylation in many cases will lead to a different association of the association of a specific protein with different complexes, and also we can see that specific splice forms will associate with different complexes in the same cell. So this is exactly going into the direction of the question from the person, and I think this is an exciting direction this research is going to progress. To not only say, well, they do associate or do not associate, but to say why, what is the molecular basis for the association, for instance, a phosphorylation site.
Excellent. We may have another question from Natalie. What is an example of a longitudinal, individual and common effect?
And a longitudinal effect would be an effect where the protein measured in a person at different time points of their life would have different variability, or more specifically in a cohort of individuals at different age. The variability would become larger or smaller as the age progresses, and that was clearly observed with quite a large number of proteins. And proteins that are affected by the environment would be proteins that when people live in different environments, they would have a different abundance distribution depending on their environment, and these sources of variation can be calculated or estimated based on this formalism that is on this slide and has been worked out by genetics community over the last decades.
Okay, I think the hour is up. I'd like to thank Ruedi and Oliver for their presentation and making this webinar possible, and I would like to thank all the listeners for listening in and asking questions. This session has been recorded and it will be available on the Biognosys website relatively soon, so you can then download it and listen to it again. I think it was a very interesting and highly up-to-date with unpublished results insight into the next generation biomarker discovery allowed by the new technologies that are developed here at the Institute and now at Biognosys as well in Zurich. So thanks very much for attending and organizing this, and hope to see you at the next webinar.
Thanks everybody.